Hey, hi guys. Welcome to online tutoring, our first online tutoring session for ANAT 212. We are going to begin by talking about blood. I'm going to start with the general characteristics of blood. Blood, the color of blood depends on how much oxygen is being carried in that blood. So as you can see here, there's bright red blood. That means that it is more highly oxygenated. And then we have this deep red purple blood here, and that means that it has less oxygen. The pH of blood is slightly alkaline. It's always, as far as the temperature, it is always slightly higher than body temperature. And as far as volume, it varies between males and females. On average, males have slightly more than females just due to body size alone. The viscosity of blood is 4 to 5 times thicker than water, and it depends on the amount of dissolved substances relative to the amount of fluid. The functions of blood is that it transports metabolic wastes, respiratory gases, and hormones. It also maintains body temperature, body pH, and fluid volume, and it protects against blood loss and infection. Okay, on this slide here, we're looking at whole blood, which once it is centrifuged, which is a machine that basically once you get your blood drawn, all of these components here are mixed. And the centrifuge right here spins it really, 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 really fast, and it causes these substances to separate. And so we can see here that in whole blood, about 55% of it is plasma. That's this stuff up here on the top. It's about 92%, so mostly water. It's got some proteins, and it's also got some other solutes like electrolytes, nutrients, respiratory gases, waste products. And then we have what we call the buffy coat, and this is just this little tiny spot right here. It's about less than 1% of the total of whole blood. And in that is platelets and some leukocytes, some white blood cells. And the other 44% of whole blood is our erythrocytes or red blood cells. We are going to now go over hemopoiesis. And hemopoiesis is the production of the formed elements, which is our red blood cells, our platelets, and our white blood cells. And so this chart kind of shows us that process depending on the type of cell. So as you can see here in hemopoiesis, we have one hemocytoblast, which is a blood stem cell. This hemocytoblast will differentiate into either a myeloid stem cell or a lymphoid stem cell. So we have our myeloid line and our lymphoid line. And as you can see, if you just kind of look down without looking at too many details, this myeloid stem line or this myeloid stem cell will then differentiate into a red blood cell, platelets, our white blood cells. So all of these come from the myeloid stem cell line. So from the myeloid stem cell line, we have erythropoiesis, which is the maturation of red blood cells or the production of red blood cells, thrombopoiesis, which is the production of platelets, and then we have leukopoiesis, and we have the leukopoiesis, which is the maturation or production of our white blood cells. And on the myeloid stem cell line, we have our granulocytes that come from the myeloid stem cell line and monocytes. And what comes from the lymphoid line here with the lymphoid stem cells are our B lymphocytes, our T lymphocytes, and our natural killer cells. So let's kind of go into a little bit more detail here with erythropoiesis. So we have our hemocytoblast, our blood stem cell, which has differentiated into a myeloid stem cell, which then differentiates into a progenitor cell. As you can see, that happens in all of these. And then it starts maturing until it becomes this red blood cell here, a erythrocyte. With thrombopoiesis, it again starts out as a progenitor cell. The thing that's really cool about platelets is then it differentiates into this megakaryoblast and then this pro-megakaryocyte, and then it turns into a megakaryocyte. And from here, this megakaryocyte then breaks up into these small pieces, which then become our platelets. Also, with our myeloid stem cell line, we have leukopoiesis along the granulocyte line and the monocyte line. The granulocyte line, these cells start as a myeloblast from the progenitor cell, and then they differentiate into eosinophils, basophils, or neutrophils, and those are all of our granulocyte white blood cells. This progenitor cell will also or can also differentiate into our monocyte, which then matures and becomes a monocyte, our white blood cell monocyte. We come back up here to this hemocytoblast, our blood stem cell. It can differentiate into a lymphoid stem cell. And from here, it can become a B lymphoblast or a T lymphoblast or continue developing into this natural killer cell here. This B lymphoblast will mature into a B lymphocyte, and the T lymphoblast will mature into the T lymphocytes. These are also white blood cells that you guys will learn more about as you get into the immune system. And as I mentioned before, erythropoiesis is red blood cell production. Go over these just one more time. Red blood cell production, platelet production is thrombopoiesis, and leukopoiesis is white blood cell production. And that is broken down into, leukopoiesis also has the myeloid line and the lymphoid. Okay, now I'm just going to touch on blood types. You guys will be doing more of this in lab. So we all have blood types. There are 4 different types. They're called ABO blood types. And the blood group depends on the surface antigen that is projecting from the red blood cell membrane. So for example, with type A, they have surface antigen A, which is represented here in these little round projections projecting from this erythrocyte. With type A blood, the plasma has anti-B antibodies. So as you can see here, this antibody is the same shape as these B antigens. With type B blood, the surface antigen here is a type B on the erythrocyte cell membrane. In the plasma of type B blood, we have anti-A antibodies, which, as you can see, fit nicely or bind to this round shape of this surface antigen A. In type AB blood, the surface antigens on the red blood cells are both A and the plasma does not have any antibody. With type O, there are no surface, either A or B surface antigens. However, in the plasma, it has both anti-A and anti-B antibodies. We also have an RH blood, and the presence or absence of this we call an antigen D. We'll determine if we are RH positive or RH negative. See, if you are RH positive, you have this surface D antigen or your RH positive because you have this specific antigen on the outside of your red blood cells. With the plasma, it does not have any anti-D antibodies. If RH negative, there are no surface antigen D. There's no D antigens on the surface of the red blood cell. And there are also no anti-D antibodies until that blood is exposed to RH positive blood. And then it builds antibodies against that blood cell. The ABO group and the RH blood type are usually reported together. For example, A positive, B negative, O positive. And it's very, very important. That's why when people give blood and people receive blood, they have to make sure that they are in match. And the reason for this is explained here. It's called an agglutination reaction. And this occurs when recipients' antibodies bind to transfused erythrocytes and clump together. And this is really serious. It can block blood vessels, prevent normal circulation, and cause hemolysis, which is the rupture of their erythrocytes, red blood cells, and it can cause organ damage. That's why it's super important. I'm going to kind of walk through it here. So here we're using the example of type A blood of the donor. It has surface antigen A, and type A blood of the recipient does not have those antibodies. It has anti-B antibodies. And so those antibodies in the recipient's blood are not going to bind to these because they can't, they're not the same shape. And then we don't have any clumping as a result. None of those red blood cells are going to stick together and we have a successful blood match. With the second scenario, we have a type A blood donor. So this person is giving blood and they have these type A surface antigens on their red blood cells. If the recipient is type B blood, they have the antibodies for type A in their plasma. And so that creates a problem. See here how here is the donor's red blood cells. Here's the recipient's antibodies from their plasma, and it's binding these red blood cells. And the result, as you can see here, is this clump, and that is what causes the problems. This is an unsuccessful blood match, and as I mentioned earlier, can cause hemolysis, block blood vessels, prevent normal circulation, and it can cause organ damage as well. That's why it's so important to make sure that those giving blood and those receiving blood are the same blood type, or that they are compatible. Okay, the last part of our review, I'm just going to briefly go over hemostasis, which is the stoppage of bleeding. There are three main steps that occur in hemostasis. The first is a vascular spasm, which is an immediate response from the body, which causes immediate vasoconstriction. It's usually in response to an injury. And then we have platelet plug formation right here. And then we have our coagulation phase, which involves the coagulation cascade. So first what happens is when we have an injury here, we get immediate vasoconstriction to limit the bleeding to that area. So we have chemicals also when they're damaged. There's chemicals that are released. These are called chemical mediators that are released from the endothelial cells and also the damaged tissue, saying, hey, we're damaged, we need help. And there's also pain initiated in this as well. But the first step is vascular spasm, and it's basically immediate vasoconstriction that happens. Then we have platelet plug formation, and here you see these platelets sticking together, and normally platelets don't stick to each other or to the inside lining of the blood vessels at the endothelium. However, when the endothelium is damaged and the underlying collagen is exposed, these little things right here are these collagen fibers. The platelets adhere to these collagen fibers right here and then start sticking together. And then those adhering platelets produce chemicals that attract even more platelets to the area. And so that's how we get this platelet plug. Then we have our coagulation phase. And this is where the blood turns from a liquid to more of a solid and a clot is formed. And this happens through the coagulation cascade. And this slide here is the coagulation cascade. We've got three pathways. We've got two pathways that then come into a common pathway. And we've got this intrinsic pathway here, our extrinsic pathway, and then they come together to form the common pathway. With the intrinsic pathway, it's initiated by damage that's done to the inside of a blood vessel. The extrinsic pathway is initiated by damage to tissue outside of a vessel. So here you can see there's injury to this blood vessel here. It initiates this intrinsic pathway. Here she's cutting something and has cut her finger. That would initiate the extrinsic pathway. And they all really work together. And so this is just basically the pathway of what happens to form this clot. In the intrinsic pathway, there are really 5 steps that kind of go along with this. The platelets will adhere to the vessel wall and release factor 12. Then factor 12 converts into inactive factor 11, and factor 11 converts to factor 9, and factor 9 to 8, all along here. This is why it's called a cascade, because all these factors into another. With the extrinsic pathway, you've got this damaged perivascular tissue, then initiates tissue factor 3. And we also have tissue factor 7, which then these can come together to initiate factor 10, prothrombin, fibrinogen. This is what starts to form the mesh and forms the clot here. That is our review for ANAT 212